Hello, welcome back to our lecture series. Emeka Udenze is speaking once again, and today we'll be talking about cellular energy and coenzymes. Quickly, let us start in the usual tradition with a food for thought or a quote. Adversity causes some men to break, others to break record. Food for thought. This quote is from William Arthur Ward. When we go through difficulty, it shapes us into becoming stronger so that we can overcome in the future. Whereas others can break down, but the ability to stay strong in the midst of adversity is what shapes us to become stronger. And in so doing, trying to overcome those challenges, men, strong men of goodwill, people who are dogged and determined to achieve, often break record. That is exactly what this is telling us. All right, let us immediately jump into the lectures. Learning objectives. We have the following learning objectives here. They include number 1, describe the role of ATP in the production and use of cellular energy. Number 2, we'll recognize the structure of some coenzymes, ATP, NADH, FADH2, and Coenzyme A. We're going to say what the full meaning of all these names are, okay? And then we'll describe the role of these coenzymes in some common metabolic pathways. So these are the 3 learning objectives that we are going to be covering in this lecture. Let's start from ATP. ATP is an acronym of adenosine 5-triphosphate. As you can see, the name adenosine has an adenine. Triphosphate, it has 3 phosphate group attached to it. It is often touted as the ultimate energy currency of the cell because it is in this form that the cell can use chemical energy that is stored in biomolecules. It is a high energy compound. Energy that is released from the catabolism of fuel molecules like carbohydrates, lipids, proteins are stored in the phosphate bond of adenosine 5-triphosphate ATP. And like I said, part of what we're going to do in this class is to recognize the structure of this ATP. I don't want you to memorize this because I'm not going to be asking you to draw this in exam, but you need to understand its basic structure. Now, it is made up of an adenine. An adenine is a nitrogenous base that is connected to a ribose sugar. This is the sugar ribose. Of course, it is a 5-carbon sugar. That's why it's the ribose. And then the ribose itself is now connected to a triphosphate. A triphosphate is 3 inorganic phosphate that is connected to each other to form this. And of course, these bonds are the phosphoester bond. This is the phosphoester bond. So this is the phosphoester bond. So this bond is the phosphoester bond. So the combination of these 3 things is what forms the ATP molecule, the adenosine triphosphate, the adenosine, which is the most important energy. That is the commonest way the cells use the energy that is stored in biomolecules. All right, we now go to how does ATP function? How does it release this energy? So hydrolysis of ATP. So the hydrolysis of ATP actually releases huge amount of energy because, now let's see, the triphosphate end, remember this triphosphate end is where that energy is stored. The triphosphate end of ATP is important in the transfer of chemical energy in the body. So the hydrolysis of those end is spontaneous. So the hydrolysis of that triphosphate end is spontaneous. When something is spontaneous, what it means is that it can occur without an input of any external factor or energy. Example, like if a ball is rolling down a hill, look at the ball. If a ball is kept in a hill and you push it down, it will roll without any input because this is what a spontaneous, this process is a spontaneous process. A ball rolling down a hill releases huge potential energy. This is exactly what happens when ATP is being hydrolyzed. So this hydrolysis results in the release of energy used to power most metabolic reactions that require the input of energy. Some reactions cannot take place and will only require energy given them by ATP for the reaction to go on. All right. So what this thing does is to push reactions that are non-spontaneous so that they can be spontaneous and actually proceed in the direction of the reaction. Now, there are two ways this hydrolysis can take place. ATP can be hydrolyzed by cleaving up just one of its ATP. Now look at what is happening here. If you use, for every hydrolysis is use water to break a bond. If water is used to break only one phosphate bond from ATP, it's going to remain ADP. ADP means adenosine diphosphate. And then the inorganic phosphate plus energy. It is this energy that is used to power metabolic activity. So when one phosphate group is released, one inorganic phosphate is released, the product becomes ADP or adenosine diphosphate. You have three, you removed one, it remains two. So this actually is the inorganic phosphate released in this case, and energy. Now, secondly, if ATP loses if it loses two phosphate groups, the two phosphate groups are usually combined together to form the pyrophosphate. This is the pyrophosphate. This is the pyrophosphate, which simply means two phosphate groups that are connected to each other. You either write it PPI or 2PI, the pyrophosphate. So when the pyrophosphate is lost, you only have one ATP, one phosphate group remaining, therefore forming the AMP. AMP simply means adenosine monophosphate. So in each of these two hydrolyses, the common thing there is that they result to the release of huge amounts of energy, and this energy is actually what is used to power metabolic reactions. When I go to the ATP ADP cycle, see, this particular cycle plays an important role in linking energy production with the energy utilization. Since ATP is what is involved in the release and production of energy and usage, so the ability of the body to see to this hydrolysis and then the reformation is what controls energy usage in the body. So look at the cycle. In the cycle here, look at what happens now. Oxidation of fuel molecule or biomolecule results to the production of what? ATP. So how does that happen? Usually what happens is that one inorganic phosphate is combined to ADP that results to ATP. So this happens when the oxidation of fuel molecules takes place, like in glycolysis, in citric acid cycle, in beta oxidation. And then what happens next? Now that ATP is now hydrolyzed. So here ATP is hydrolyzed. Hydrolysis or ATP results. So when you hydrolyze ATP and cleave off that phosphate group, you form your ADP back. So you recycle your ADP back, whereas this results to what? Plus energy. And that energy released is used to do cellular work or biosynthesis. The cellular work includes movement of the muscle and, let's say, active transport. And then biosynthesis means synthesis of biomolecules. So the energy released in the hydrolysis of this ATP is actually used in doing cellular work and biosynthesis. And this hydrolysis results to this release of energy, which is used to do this work. So here, this hydrolysis results to what? Hydrolysis results to release of energy. Release of energy to power metabolic work and cellular work as well. We now go to some important fun facts about ATP that we need to know. ATP is produced from the catabolism of nutrients of the fuel molecule, the carbohydrate, lipids, and proteins. Now, a resting human body hydrolyzes ATP at the rate of 40 kilograms. You see, that's a lot. Every 24 hours. Because your body, every work done by the cells depends on this. Now, most of the ATP synthesis occurs in the mitochondria. Or if it is singular, you can say mitochondrion. Remember that. If it is one, you can say mitochondria. So the mitochondria contains those enzymes of the citric acid cycle of the beta oxidation, where they help in facilitating the processes, the breaking down of biomolecules that result toward the formation of ATP. A few of them occurs in the cytoplasm. In that case, we call it the substrate level phosphorylation. We're going to talk about that in the next lecture. Now, ATP molecule is hydrolyzed within 1 minute following its formation in the typical cell. In a typical cell, the moment ATP is produced, it is what? Broken down and used to provide energy and converted back to ATP. These are important form facts you need to know. And this kind of thing, I can ask you an exam, particularly in a multiple choice question. Coenzyme A. Now let's go to the second one we need to look at, the coenzyme A. The coenzyme A is a central compound in the metabolism. Is a central compound in metabolism and forms part of the very popular common intermediate, the acetyl-CoA. Remember when we talked about the different levels, stages of extraction of energy, we said at a point, every molecule is converted to a common product in stage 2, which is acetyl-CoA. So it's, when we say acetyl-CoA, the CoA part, the CoA is what we're talking about, is the coenzyme A. The CoA is what we mean by the coenzyme A. Now, it is derived from the vitamin B. The vitamin B is what? The pantothenic acid, and contains a phosphate group. You see, it contains a phosphate group that is derived from adenosine diphosphate ADP, a beta mercaptoethylamine, and then a reactive sulfhydryl, or what we call the thioester, the thiol group. We see the thiol group. It contains the thiol group. So let's look at the structure now. Okay, let's start from here. Now this is, remember this is of course is adenine. Then adenine itself is connected to a ribose. Ribose is now connected to 2 phosphate groups. So if you look at this, this looks like a derivative of ADP. So if you remove this guy now, if I do, assuming I remove this one, this and this forms adenosine diphosphate ADP. However, if you now put this one here, we say it's a derivative of ATP, just to let you know. And then this is now connected to the pantothenic. This is the pantothenic acid, the B vitamin, pantothenic acid, vitamin B5. And then this is now connected to this compound, this beta mercaptoethylamine. This ethylamine is connected. Now, look at what matters. The reactive group is on this side. It is here that connects itself to an acyl that forms a type of acyl-CoA. And when it is an acetyl-CoA, when it's an acetyl, we call it acetyl-CoA. So this is the reactive point. So it is this point that connected to the C group. This is connected to a C group. A C group is connected to a C group. And like I said again, or acetyl-CoA. Now let's see what happens there. So its main function is it transfers acyl groups. It transfers what's an acyl group? Acyl groups are, look at an acyl group. Acyl group simply means one carbon is a carbonyl carbon connected to any carbon chain. We call it an acyl group. And then when it is now connected to the acetyl-CoA, look at it now. It is now connected to the CoA. This is the CoA. Remember, there's supposed to be SH here, but that H is gone. You're now connected to this. This forms the thioester bond. So this is the CoA part. This is the acyl part. So this is what we call the acyl-CoA. Now, if this acyl-CoA is acetyl, it becomes acetyl-CoA like you have in this case. So what we have done is that this is 1. So if you count this 1, 2, just 2 carbon with the carbonyl carbon here, we call this the acetyl. And the name of this now becomes acetyl-CoA. So it can be any other, it can be 3, it becomes propionyl-CoA. If it's propionate, it becomes propionyl-CoA, right? Butynyl-CoA. So here, because it is 2, we call it what? Acetyl-CoA. So what it simply does is that it transfers acyl groups. That's what it does. So this acyl group connected here can now be transferred to any other substrate or intermediate in reaction. So the acyl groups are linked to the CoA via a sulfur atom in a thioester bond. Like I said again, this is the thioester bond. And that is the main function of the coenzyme A. We now go to the next one. The nicotinamide adenine dinucleotide, reduced form. I purposely put the reduced form. Remember, we define reduction as addition of hydrogen, whereas oxidation is the removal of hydrogen. That's it. So this is the reduced form because it has a hydrogen to it. So the oxidized form usually exists as NAD+, remove without a hydrogen. So it is derived from ADP again, and then vitamin B3, the nicotinamide. Look at it here. So this is, let's see, this is adenine to a ribose to 2-phosphate, diphosphate. So from here to here is the ADP. So if you go from, let's see, This is ADP. And then connected to another ribose. It is this ribose that connects these to the nicotinamide ring. The nicotinamide ring, fortunately, is the point of the reaction site. So the reaction site is located at the nicotinamide ring at this point. And what does it do? It acts as an electron donor or proton or hydrogen donor, whichever you want to use. So nicotinamide ring accepts 2 protons. That's what it's 2 protons. Sorry, 2 electrons rather. It accepts two electrons and 1 proton. It's important we know that it accepts two electrons and 1 proton during oxidation of the substrate, and now forms what? The reduced coenzyme, which is NADH. So look at what happens here. So if this is NAD+, which is the oxidized form, this is the oxidized form, like I told you, without hydrogen, it accepts hydrogen. So one of them gets here to form the reduced one, plus one of the hydrogen now begins to flow. So this is reduced, and this is oxidized. This is the reduced, and this is the oxidized form. So, and the reaction center is at this point. So this hydrogen can be removed or added. If you remove it, it becomes NAD+. If you add it, it becomes NADH. And that's the main. So its main function is to transfer that hydrogen or proton between substrate and its intermediate. We now go to the next, to its typical reaction. A typical reaction of nicotinamide adenine dinucleotide. So a generic one is here. Look at what happens here. This is an alcohol. And this alcohol, look at what happens. This alcohol loses these 2 hydrogen. This alcohol loses a hydrogen here and a hydrogen here. That's exactly what it does. So those 2 hydrogens are lost. If you lose these 2 hydrogen and transfers it together, look at what happens. This one now gets one of these to become NADH, and one of the hydrogen ion is now. So anytime NADH is reduced, one of the hydrogen ions actually float about, whereas one of them gets attached to it. Whereas the molecule now loses them. So look at what happens here. The moment this loses this, it forms a double bond, a carbon-carbon, and one of them goes off. So here now, since this has more hydrogen, this is the reduced form of the substrate. This less hydrogen, this is what? The oxidized form. So this is a typical reaction that NADH gets itself involved as a coenzyme. And enzymes that do this are usually called dehydrogenases because they remove or add hydrogens. That's what we call it, a dehydrogenase. So in this case, this is an alcohol. We can actually name this alcohol dehydrogenase. That's exactly what we're going to name this. We now go to the next one, the flavin adenine dinucleotide. Flavin adenine dinucleotide, FAD. In this case, this is the oxidized form. So the reduced form is, the reduced form is FADH2. Now it has 2 hydrogens, making it reduced. So it is derived again from ADP and vitamin riboflavin. Vitamin B2 is the riboflavin. Now the reaction site is located on the riboflavin ring, and in this case, what it does is that it accepts 2 electrons or 2 protons. It accepts 2 full protons to become part of the reduced form. Let's see what happens. So again, this is adenine connected to a ribose and connected to a diphosphate. So this is the ADP part. Of course, from here is the ADP. From here is the ADP part. Well, let me make that look good. Yeah, this is the ADP part. And then connected to the right, see, this is the riboflavin part. From here to here is the riboflavin part. And on this ring is where the reaction takes place. So look here, hydrogen is now added on both of these sides to make it reduced or removed to make it oxidized. So it accepts 2 hydrogens, unlike the NADH that only accepts 1 hydrogen, allowing the other hydrogen to float around. Let's see a typical reaction where this takes place. So substrate of enzyme that use FAD or FAD, flavin adenine dinucleotide, as coenzyme give up 2 electrons, whereas the FADH itself does what? Accept 2 hydrogens or 2 protons. It is involved in reactions in which single bonds part of the substrate is converted to double bond. Let's see what happens here. Now, this is a typical saturated compound. Look at it. It is highly saturated, single bond. Now, these two hydrogens is going to be lost. The moment you lose these two hydrogens, it gets saturated and forms a double bond. Look at what happens here. It loses the two hydrogens and forms a double bond. It becomes unsaturated. Remember, if it is saturated, more hydrogen means is reduced. Here, unsaturated, it becomes oxidized. Look at where does that hydrogen go? It goes to this guy. So both of these hydrogen goes to this guy to become FADH2. And this is what it does. And of course, this is what? The reduced form. And of course, this is what? The oxidized form. And this is exactly how FAD, or flavin-adenine dinucleotide, performs its role in metabolism. We now go to the next one. Okay. I think having said that, we've actually come to the end of this lecture with the mechanism of action of flavin-adenine dinucleotide. Thank you very much for listening. Bye at this point.